Hello everyone, welcome back. This is Biochemistry Metabolism and we are in lesson 5. And in this lesson we're going to talk about glycolysis regulation in the liver. So last time we talked about the muscle and we learned about the different types, and this time we're going to apply them here as well. See how is it different in the liver and how is it similar. And let's get into it. So remember, we have 3 irreversible steps in glycolysis. So step number 1, right? Step number 3. Then I've skipped a bunch here. And we go straight ahead to step number 10, where it's irreversible. So let's start with 1, and we're going to work our way down. So firstly, hexokinase. In the liver, you have a different isoform of it. And in the liver, it's called glucokinase. And this glucokinase is actually not inhibited by glucose 6-phosphate. Remember, in the muscle, glucose 6-phosphate inhibits hexokinase, right? Negative feedback. In the liver, you don't have that. And why do you not have that? Well, because glucokinase has 50-fold lower affinity. For glucose than hexokinase in the muscle. Therefore, regulation is not necessary. It will only work when there are high levels of glucose. So it works when glucose is high. And that is important because the liver doesn't want to take glucose, right? It wants to give it to other tissues, like the brain, where it really needs it. Therefore, glycolysis in the liver happens when you have glucose in the body. Because if you don't have glucose, right, in the blood, what your liver does is it goes through gluconeogenesis to produce glucose. And nothing else affects hexokinase or glucokinase in the liver. Therefore, we're going to move on to PFK-1. Now, as we said before, PFK-1 is the committed step. So this is the committed step for glycolysis. Why? Because glucose 6-phosphate is the intermediate for other metabolic pathways, while PFK-1 is the first intermediate where PFK-1, the reaction with PFK-1, fructose 1,6-bisphosphate is the intermediate that is used for glycolysis only, right? It does not go to other pathways unless you switch to gluconeogenesis, and it could go backwards with the bypass enzyme we've mentioned before. And in this committed step, in the liver, right, unlike ATP needs, you do not care about ATP needs. Because in the liver, ATP isn't necessary. You don't have insane surges or plummets of ATP, right? So ATP is not a form of regulation. So in the liver, ATP is not a form of regulation, right? ATP ratio. So let's say ATP to AMP ratio. Well, it's not. However, in the liver, we have citrate. We also have citrate in other tissues because it is a product or an intermediate from the Krebs cycle. However, in the liver, citrate leaves the mitochondria, it goes to the cytoplasm where glycolysis takes place, and what it does is it inhibits PFK-1, right? So it inhibits. And why does it inhibit? Well, it's a signal that the cell has metabolic needs met, right? The cell has enough nutrients or enough molecules down the path that they don't need it anymore. So excess levels of citrate, they go and they inhibit PFK-1, meaning no need to produce anything else. We have enough downstream. Another molecule that acts on PFK-1 is fructose 2,6-bisphosphate. So this isn't the same as fructose 1,6-bisphosphate. And what this does is it activates PFK-1. So you have activation. And we're going to go more into it later on. Let's move on to pyruvate kinase, and we'll go back to fructose 2,6-bisphosphate because that is kind of the main mechanism of liver regulation concerning maintaining blood glucose levels. Moving on to pyruvate. Firstly, pyruvate is activated by fructose 1,6-bisphosphate. So similar to the muscle, you have this activation. So what happens is this activates it. So it's feed-forward stimulation. We've talked about this before. Something else that happens here is alanine, right? You have alanine. It's an amino acid. What does it do? Well, it inhibits, right? Inhibits because it's a signal for having enough metabolic needs. So alanine works in a similar fashion to citrate. Something else that works on pyruvate kinase, and this also happens in the muscle. I did not mention it in the last video. I'm mentioning it now because it kind of happens more in the liver, but you can consider it happening in both the muscle and the liver, and that is phosphorylation of pyruvate kinase. Now, pyruvate kinase is active in its unphosphorylated form. So when you phosphorylate it, so if we phosphorylate it and we get, so let me write it with blue, we get pyruvate kinase and it's phosphorylated. So it's phosphorylated. That's better. That's better. Alright. So it has a covalent modification. And what this means is that it's inactive now. So this is its inactive form. So it's inactive. And here it's active. And how do we get to its active form? Well, we dephosphorylate it. So you release it. And we actually add in water here. We also use ATP. And we phosphorylate it, and then it's dephosphorylated to pyruvate kinase when the cell needs to. And this happens in the muscle as well. So we have the covalent modification and we have some allosteric modifications. And now we're going to talk about some hormonal and covalent sort of regulation for fructose 2,6-bisphosphate, which eventually acts on PFK-1, thus stimulating it. And this is a more complex mechanism. Let me go more to the right. Alright, so first things first, I'm going to draw two enzymes. And this one first is PFK-2. So we have phosphofructokinase 1, right? That's the one that fructose 2,6-bisphosphate acts on. And phosphofructokinase 1 catalyzes the third step in glycolysis. PFK-2 is used in regulation of PFK-1. And this second enzyme, and together they're a bifunctional enzyme. So we can say bifunctional enzyme. And the second one is a fructose bisphosphatase. Alright, perfect. So this enzyme regulates fructose 2,6-bisphosphate. And how is that done? So we're going to assume a few things here. So firstly, let's assume you have fructose 2,6-bisphosphate, right? It's activating PFK-1, you have glycolysis. This means you have energy, right, to use. However, let's say you run out of glucose in the blood. You want to switch to gluconeogenesis and you want to reduce glycolysis, right? So let's say you want to increase gluconeogenesis. And you want to reduce glycolysis because you have low amount of glucose in the blood, right? Which hormone tells you have low amounts of glucose in the blood? Well, that's glucagon. And glucagon does a signaling pathway, which eventually activates a kinase that is known as protein kinase A. And you must have heard of this. It's a very common pathway. It's one of the first ones you would learn if you're getting into biochemistry. And what this does is it catalyzes a phosphorylation. So you have pKa, right? Protein kinase A, and you use ATP, and you phosphorylate it, and you get the phosphorylated form of this bifunctional enzyme. So we're gonna have PFK-2. Oh, and this is fructose bisphosphatase 2. Forgot to mention, it's 2 because we have fructose bisphosphatase PFK-1 in gluconeogenesis pathway. And fructose bisphosphatase 2. There's no space. Anyways, so here it's phosphorylated. I'm going to add a phosphate group. Alright, perfect. This phosphate group, the addition of it, it's somewhere on the enzyme. Don't assume this is the accurate location. What this does is it inactivates PFK-2. So the phosphorylation status of this bifunctional enzyme inactivates PFK-2. So PFK-2 is inactivated. So therefore fructose bisphosphatase 2 is activated. And what this does is, because you have low amount of glucose in the blood, right? We got that because of glucagon. Now you want to activate gluconeogenesis, therefore inhibiting PFK-1. And how do you inhibit PFK-1? By reducing fructose 2,6-bisphosphate. And how do you reduce it? Well, you break it down. You break the phosphate and you get fructose 6-phosphate. And that's done by fructose bisphosphatase 2. So what that does is you have fructose 2,6-bisphosphate, right? And what this does is it cleaves the phosphate. So you have the release phosphate, and you get fructose 6-phosphate. And this can go and become glucose if it wants to, can do whatever it wants. However, now we have less fructose 2,6-bisphosphate, therefore glycolysis is less active as we wanted. Now let's assume the opposite. We have high amounts of glucose. You just ate, your body, your blood vessels are filled with glucose. What happens? Insulin is secreted, right? By the pancreas. Now insulin binds on its receptor, which is a tyrosine kinase, and due to its binding, you eventually get a phosphatase active. And this phosphatase, that's known as phosphoprotein phosphatase. It cleaves the phosphate group. So now you have PFK-2 and fructose bisphosphatase 2 without the phosphate group. And in this case, PFK-2 is active, fructose bisphosphatase 2 is inactive. And you guessed it. What does PFK-2 do? Well, it does the opposite. What do? Well, you have fructose 6-phosphate, right? And you have a lot of it because you have high amounts of glucose, which converts to fructose 6-phosphate, right? Because you get glucose to glucose 6-phosphate and glucose 6-phosphate to fructose 6-phosphate. And as fructose 6-phosphate, not only does it convert to, fructose 2,6-bisphosphate, it can also activate. It can also activate this, right? It activates phosphoprotein phosphatase so that you get more PFK-2 and more of it is converted into fructose 2,6-bisphosphate. And what does this fructose 2,6-bisphosphate do? Well, we talked about it before. It goes and activates phosphofructokinase 1. Therefore, you have glycolysis active. And therefore, in this case, I'm going to write you have an increase in insulin levels. Right? Therefore, you have less gluconeogenesis. And you have an increased amount of glycolysis. And this is all logical, right? So why is insulin higher? Because you have high amounts of glucose in the bloodstream, right? And why do you want to reduce gluconeogenesis? Because you have high amounts of glucose in the body. And therefore, glycolysis is active because you have high amounts of glucose in the body. And let's say glucose runs out. What happens? You have less secretion of insulin, more secretion of glucagon, and you switch the pathway. And you break down fructose 2,6-bisphosphate. So this is broken down. So you do this pathway. You break it down. You get fructose 6-phosphate. And this fructose 6-phosphate, right, does not activate PFK-1. You have less activation of PFK-1 because fructose 2,6-bisphosphate is broken down. And therefore, you switch to gluconeogenesis to eventually get glucose. And this is kind of the main way the liver regulates glycolysis by affecting PFK-1. Therefore, PFK-1 can know the amount of glucose in the blood and act with it. So a high amount of glucose, it's activated. It does glycolysis. Low amounts, it's not activated, and now the cell can do gluconeogenesis, and the liver can produce glucose for the rest of the body to use.